Bioluminescent Imaging in Neuroscience, Unraveling Brain Circuitry. Title: Bioluminescent Imaging in Neuroscience, Unraveling Brain Circuitry. Introduction. The human brain is a complex network of billions of neurons, intricately connected to form circuits that give rise to our thoughts, emotions, and behaviors. Understanding the complex wiring and functionality of these neural circuits has been a long-standing challenge in neuroscience. Over the years, scientists have developed various imaging techniques to gain insights into the inner workings of heat brain. One such technique that has emerged as a powerful tool is bioluminescent imaging. The basics of bioluminescence. Bioluminescence is a fascinating natural phenomenon exhibited by a wide range of organisms, including bacteria, fungi, marine creatures, and fireflies. It is the process through which these organisms produce and emit light. This natural light emission stems from chemical reactions within the organism, involving the enzyme luciferase and a luciferin molecule. The luciferase enzyme acts as a catalyst, converting the luciferin into an excited state, which eventually decays and releases energy in the form of light. By genetically engineering the luciferase enzyme into neurons or other targets, say LLS, researchers can effectively tag these cells with bioluminescence, allowing for optical detection and imaging. Advantages of bioluminescent imaging. Traditional imaging techniques such as functional magnetic resonance imaging (FMRI) and positron emission tomography (PET) have been foundational in understanding brain activity. However, these techniques often suffer from limitations such as low spatiotemporal resolution, invasiveness, or the need for radioisotopes. In contrast, bioluminescent imaging offers several advantages that make it particularly well suited for studying brain circuitry. Firstly, the localization and quantification of bioluminescent signals are highly precise. This allows researchers to track the activity of specific neuronal populations within intricate brain circuits, providing insights into neural communication and information flow. Secondly, compared to other imaging techniques, bioluminescent imaging has excellent temporal resolution, enabling the detection of rapid changes in neuronal activity. This advantage is particularly critical when studying processes that occur on a millisecond timescale, such as synaptic transmission or the dynamic interactions between different brain regions. Thirdly, bioluminescent imaging is non-invasive, making it suitable for longitudinally monitoring brain activity over extended periods. This enables researchers to observe how neural circuits evolve in response to neurodevelopment, learning, or disease progression. Applications of bioluminescent imaging in neuroscience. The use of bioluminescent imaging has revolutionized our understanding of brain circuitry and contributed to numerous breakthroughs in neuroscience research. One of the prominent applications is in mapping brain connectivity, whereby researchers use bioluminescence to track the flow of signals within and between different brain regions. Bioluminescent imaging has also made significant contributions to the study of neuroplasticity, the brain's ability to change and reorganize in response to experience. This insight into plasticity mechanisms can lead to valuable interventions for conditions such as neurodevelopmental disorders and cognitive decline. Moreover, bioluminescent imaging has proven useful in studying brain disorders and exploring potential therapeutic interventions. This information can help identify critical nodes within dysfunctional circuits and guide the development of targeted treatments. Future directions and challenges. While bioluminescent imaging has undoubtedly transformed our understanding of brain circuitry, several challenges and future directions remain. For instance, improving the detection sensitivity in Signal to noise ratio is critical for achieving higher resolution and more accurate imaging. Advances in genetic engineering, such as the development of brighter luciferase enzymes or luciferin variant (TS), will be instrumental in overcoming these challenges. Additionally, the application of bioluminescent imaging to larger animals, including non-human primates, will be essential for better modeling the complexities of the human brain. This expansion will allow for the translation of preclinical findings into potential clinical applications, providing hope for novel diagnostic and therapeutic strategies. Bioluminescent imaging has transformed our understanding of brain circuitry, offering neuroscientists unprecedented insights into the complex wiring and functionality of neurons. This technique harnesses the natural light emitted by various organisms, allowing researchers to study the brain with remarkable precision and non-invasiveness. Bioluminescence is a fascinating natural phenomenon exhibited by bacteria, fungi, marine creatures, and fireflies. It involves the production and emission of light through chemical reactions within T. organism, utilizing the luciferase enzyme and luciferin molecule. By genetically engineering the luciferase enzyme into neurons or target cells, researchers can tag these cells with bioluminescence, enabling optical detection and imaging. Compared to traditional imaging techniques, bioluminescent imaging offers several advantages for studying brain circuitry. Firstly, it allows for highly precise localization and quantification of signals, enabling researchers to track the activity of specific neuronal populations within intricate brain circuits. This provides valuable insights into neural communication and information flow. Secondly, bioluminescent imaging has excellent temporal resolution, making it possible to detect rapid changes in neuronal activity. This is particularly valuable for studying processes that occur on a millisecond timescale, such as synaptic transmission or dynamic interactions between different brain regions. Thirdly, bioluminescent imaging is non-invasive, allowing for longitudinal monitoring of brain activity over extended periods. This provides researchers with the opportunity to observe how neural circuits evolve in response to neurodevelopment, learning, or disease progression. The applications of bioluminescent imaging in neuroscience have been significant. One prominent use is mapping brain connectivity, where researchers track the flow of signals within and between brain regions using bioluminescence. Bioluminescent imaging has also contributed to the study of neuroplasticity, observing how the brain changes and reorganizes in response to experience. This understanding of plasticity mechanisms may lead to interventions for neurodevelopmental disorders and cognitive decline. Furthermore, bioluminescent imaging has played a role in studying brain disorders and exploring potential treatments. By engineering bioluminescence markers into specific neuronal populations associated with disease states, researchers can investigate their behavior and interactions within the context of pathology. This information helps identify critical nodes within dysfunctional circuits and guide the development of targeted therapies. Despite its transformative impact, bioluminescent imaging faces challenges and future directions. Improving detection sensitivity and signal-to-noise ratio is crucial for achieving higher resolution and more accurate imaging. Advances in genetic engineering, such as brighter luciferase enzymes or luciferin variants, will be essential in overcoming these challenges. Additionally, expanding the application of bioluminescent imaging to larger animals, including non-human primates, is vital for better modeling the complexities of the human brain. This would facilitate the translation of preclinical findings into potential clinical applications, offering hope for novel diagnostic and therapeutic strategies. Bioluminescent imaging has revolutionized the study of brain circuitry in neuroscience. By harnessing the natural light emitted by certain organisms, researchers can visualize and study the intricate wiring and functionality of neurons with remarkable precision and non-invasiveness. Bioluminescence is a fascinating natural phenomenon found in various organisms, including bacteria, fungi, marine creatures, and fireflies. It involves the production and emission of light through chemical reactions using the luciferase enzyme and luciferin molecule. By genetically engineering the luciferase enzyme into neurons or target cells, researchers can tag these cells with bioluminescence.